In 1947, a Bedouin shepherd boy threw a rock into a cave near the Dead Sea and heard the sound of something breaking. When he climbed inside to investigate, he found ancient clay jars containing leather scrolls wrapped in linen. Those scrolls would turn out to be the most significant archaeological discovery of the 20th century, the Dead Sea Scrolls, the oldest known biblical manuscripts in existence, dating back over 2,000 years. For 70 years, scholars studied these scrolls, debated their origins, and developed theories about who wrote them. They analyzed handwriting styles, compared linguistic features, and examined the physical appearance of the parchment. They used every tool available to traditional archaeology and textual scholarship to understand where these precious manuscripts came from and what they meant. The consensus view that emerged shaped how we interpreted the texts, how we understood early Judaism, and how we viewed the development of Christianity. It was a theory built on decades of careful scholarship supported by the best minds in biblical studies and archaeology. Then, in 2020, a team of scientists published the results of a study that seemed impossible. They had done something no one had attempted before. They had extracted and sequenced ancient DNA from the Dead Sea Scrolls themselves, not from the people who wrote them, but from the animal skins the scrolls were written on. What that DNA revealed would stun the archaeological world and force scholars to reconsider everything they thought they knew about these ancient texts. This is the story of how molecular biology intersected with biblical archaeology in the most unexpected way. How scientists figured out how to read genetic information from parchment that is 2,000 years old without destroying the sacred texts written on it. And how tiny fragments of ancient DNA answered questions that 70 years of traditional scholarship could not definitively resolve. The Dead Sea Scrolls consist of roughly 25,000 fragments representing about 1,000 different manuscripts. They were discovered in 11 caves near Qumran between 1947 and 1956. The texts include the oldest known copies of biblical books, sectarian religious writings, community rules, hymns, prayers, and commentaries. They date from approximately 250 BCE to 68 CE, spanning a crucial period in Jewish and early Christian history. When the scrolls were first discovered, they were remarkably well preserved for their age, protected by the dry desert climate and sealed in clay jars. But 70 years of handling, study, and exhibition have taken their toll. The scrolls are now extremely fragile. Some fragments are so small and damaged that scholars can barely read them. And because the scrolls are considered sacred texts by multiple religious traditions, any destructive testing has been strictly forbidden. The sheer scale of the challenge facing scholars was staggering. 25,000 fragments, most of them tiny pieces of parchment with just a few words or partial lines of text visible. No titles, no page numbers, no clear way to tell which fragments belong to which manuscripts. It was like trying to assemble 1,000 different jigsaw puzzles that had been mixed together with most of the pieces missing and no pictures to guide you. Scholars had to rely on educated guesses based on handwriting analysis, comparing scripts to determine if fragments might have been written by the same scribe. They examined the physical characteristics of the parchment, its color, texture, thickness, hoping to match fragments that came from the same animal skin. They analyzed the content, looking for textual connections that might indicate fragments belong to the same work. This was extraordinarily difficult and risky work. A wrong match could distort scripture or ancient tradition. If you placed two fragments together that did not actually belong to the same manuscript, you could create false readings that would mislead every scholar who relied on your reconstruction. You could attribute ideas to ancient authors that they never wrote. You could create hybrid texts that never existed. The margin for error was enormous, and the consequences of error were severe. Yet scholars had no choice but to proceed with these methods because they had nothing else to work with. The dominant theory that emerged from decades of painstaking work was that the scrolls belonged to the Essenes, a Jewish sect described by ancient historians Josephus and Pliny the Elder. According to that theory, the Essenes lived in a monastic community at Qumran, isolated from mainstream Jewish society, following strict purity laws and copying sacred texts. When the Roman army approached during the Jewish revolt in 68 CE, The Essenes supposedly hid their library in nearby caves where it remained until 1947. This theory explained many features of the scrolls. The sectarian writings describing community rules fit with what ancient sources said about the Essenes. The text showing a solar calendar different from the lunar calendar used in Jerusalem suggested a group that had broken from the temple establishment. The apocalyptic texts expressing hostility toward the Jerusalem priesthood matched the image of a radical separatist group waiting for divine intervention. For decades, this became the accepted explanation taught in universities, presented in documentaries, and described in museum exhibitions. The Dead Sea Scrolls were the library of the Essenes. Case closed. But there were problems. Problems that nagged at scholars even as the Essene theory became dominant. The scrolls showed significant textual variation, different versions of biblical books that did not match each other. Some fragments were written in different scripts by different scribes using different writing conventions. The parchment quality varied enormously from fine, carefully prepared skins to rough, poorly processed materials, and radiocarbon dating suggested that some scrolls were older than the Qumran settlement itself. Most puzzling was the sheer diversity of the texts. If these were the private library of one isolated sect, why would they possess multiple conflicting versions of biblical books? Why would they preserve texts representing theological positions they disagreed with? Why would their library contain such variety if they were copying texts for their own use according to their own theological perspective? These questions troubled scholars, but without hard evidence to support alternative theories, the Essene hypothesis remained dominant. It was the best explanation available given the tools and methods of traditional archaeology and textual criticism. Then in 2012, something extraordinary happened that would eventually change everything. A molecular biologist named Oded Rechavi was having lunch with a biblical scholar named Noam Mizrahi at Tel Aviv University. They were discussing the Dead Sea Scrolls and the challenges of matching fragments and determining origins. Mizrahi explained the difficulty scholars faced trying to reconstruct manuscripts from thousands of tiny pieces with limited clues. He described the risks of wrong matches and the frustration of questions that traditional methods could not answer. Rechavi, whose specialty was genetics and DNA analysis, asked a simple question that would spark a revolution in Dead Sea Scrolls research. Rechavi asked, "What are the scrolls made of?" Mizrahi replied, "Parchment, animal skin." Rechavi paused, thinking. Then he said something that would lead to one of the most important breakthroughs in biblical archaeology. Then the parchment has DNA. It was one of those moments where two different fields of expertise intersected and created a completely new approach. Mizrahi understood the textual problems that had puzzled scholars for decades. Rechavi understood the biological solutions that might address those problems. Together, they realized they could use ancient DNA analysis, a technique that had revolutionized archaeology in recent years by revealing human migration patterns and population history, to answer questions about the Dead Sea Scrolls that traditional methods could not resolve. But there was an enormous challenge that made this idea seem almost impossible to execute. The Dead Sea Scrolls are sacred texts revered by multiple religious traditions. They are also incredibly fragile after 2,000 years of aging and 70 years of study. Any technique that required cutting samples from the scrolls, drilling cores, or removing material would be absolutely forbidden. The scrolls had to be preserved intact for future generations. How do you extract DNA from ancient manuscripts without damaging them? How do you gather enough genetic material for analysis while meeting the strict ethical and conservation requirements for handling sacred texts? The team that eventually formed around this idea included Rechavi, Mizrahi, and Penina Shor, curator of the Dead Sea Scrolls at the Israel Antiquities Authority. They spent years developing a methodology that could extract sufficient genetic material for analysis while respecting the absolute prohibition against damaging the scrolls. The breakthrough came when they realized they did not need to cut samples from the parchment itself. Over decades of handling and study, tiny particles of parchment had accumulated as dust in the containers where scroll fragments were stored. This dust contained collagen, the protein that makes up animal skin, and embedded within that collagen was ancient DNA. By carefully collecting this dust from blank margins or damaged areas where no text existed, They could gather enough material for genetic analysis without touching any written portion of the scrolls. It was non-invasive sampling at its most ingenious, using material that would otherwise be discarded or ignored to unlock secrets that had been hidden for 2,000 years. But extracting usable DNA from ancient parchment presented extraordinary challenges. The parchment-making process itself, soaking, scraping, stretching, and drying animal skins, damages DNA at the molecular level. Then the skins were written on with iron-based inks that are chemically corrosive, further degrading genetic material. Then they sat in desert caves for 2,000 years, exposed to temperature fluctuations, humidity changes, and mineral deposits from the cave environment. And then they were handled by countless people over 70 years of study, each person potentially leaving traces of modern DNA. The result, the DNA they were trying to extract, was fragmentary, severely degraded, and potentially contaminated by modern genetic material from everyone who had ever touched the scrolls. The team had to work in specialized ancient DNA laboratories with the strictest contamination controls imaginable. Everyone wore full protective gear, including suits, masks, and gloves. The lab maintained positive air pressure to prevent outside air from entering. All equipment was sterilized with ultraviolet light between uses. Work surfaces were bleached repeatedly, and every step of the process was documented and controlled. They focused on two types of genetic material: mitochondrial DNA, which is more abundant in ancient samples because each cell contains hundreds of mitochondria, gave them enough material to work with, despite the degradation. Each mitochondrion has its own small genome, so even badly degraded samples often retain some mitochondrial DNA. Nuclear DNA, though much harder to extract from ancient samples, provided more detailed information about individual animals and their relationships. The team developed strict authentication protocols to prove the DNA they were sequencing actually came from the ancient parchment and not from modern contamination. They looked for damage patterns characteristic of ancient DNA, specific types of molecular breakdown that occur over centuries. They checked for consistency across multiple extractions from the same fragment. They compared results from different laboratories to ensure reproducibility. They used sophisticated statistical methods to distinguish ancient sequences from modern contaminants. It took years of painstaking work, hundreds of samples, thousands of hours of laboratory analysis, careful validation of every result, but eventually they had reliable genetic sequences from dozens of Dead Sea Scroll fragments. And what those sequences revealed would force scholars to reconsider 70 years of established theory about the origins and nature of the Dead Sea Scrolls. The first major discovery from the DNA analysis seemed simple on the surface, but its implications were profound. When the team sequenced genetic material from different scroll fragments, they found that most tested fragments were made from sheepskin. Sheep were common in ancient Judea, and sheepskin was the standard material for making parchment. But some fragments were made from cowskin. Cow parchment changed everything. Qumran, where the scrolls were found, sits in the Judean desert near the Dead Sea, one of the harshest, driest environments in the entire region. It is a landscape of rock, sand, and sparse vegetation. Water is scarce and grazing land is minimal. Sheep and goats can survive in this environment because they are adapted to desert conditions. They require relatively little water, and they can graze on the tough, sparse plants that grow in arid regions. Cattle cannot survive in that environment. Cows need abundant water, dozens of gallons per day per animal. They need substantial grazing land with grass and vegetation. They are not desert animals. They require the resources of wetter, more fertile regions. There was no way a community living at Qumran could have raised cattle. They could not have had regular access to cowhides for making parchment. The environment simply could not support it. That meant any scroll fragment written on cow parchment came from somewhere else with water and pasture outside the desert. Somewhere with resources the Judean desert did not provide. The genetic evidence proved beyond any possibility of doubt that not all the Dead Sea Scrolls originated at Qumran. This simple biological fact, cow versus sheep, demolished a key pillar of the theory that the scrolls were the private library of one sect living in isolation at Qumran. But the DNA revealed far more than just species identification. By analyzing genetic relationships between different parchment samples, the team could determine whether multiple fragments came from the same individual animal or from closely related animals. This created a genetic fingerprint for matching fragments that might belong to the same manuscript. This was revolutionary. For the first time, scholars had an objective scientific method for determining whether fragments belong together. Instead of relying on subjective judgments about handwriting or educated guesses about parchment appearance, they could use genetics to prove connections. One of the most significant findings involved the Book of Jeremiah. The Dead Sea Scrolls preserve two dramatically different versions of this biblical book. One version is similar to the Hebrew Bible we know today, longer with chapters in a familiar order. The other version is significantly shorter, missing entire sections, and has chapters arranged in a completely different sequence. Scholars had debated for decades what this meant. Did these represent different textual traditions that developed in different communities, or were they just variant copies from the same tradition, with the differences representing scribal choices or errors? The question mattered enormously for understanding how biblical texts developed and were transmitted in the ancient world. The DNA provided a definitive answer. Fragments of the shorter version of Jeremiah were written on cow parchment. Fragments of the longer version were written on sheep parchment. They came from different animals, almost certainly from different regions given the environmental constraints on cattle. This strongly suggested they represented genuinely different textual traditions that had developed in separate communities. The differences were not just copying variations. They reflected distinct versions of the text that were being preserved and transmitted independently in different geographic locations. Another crucial test involved the Songs of the Sabbath Sacrifice, a liturgical text describing angelic worship in the heavenly temple. Copies of this text were found both in the Qumran caves and at Masada, the fortress where Jewish rebels made their last stand against Rome in 73 CE. Did the Masada text come from Qumran originally? Were all copies derived from one source? The DNA analysis showed that multiple fragments of this text found at Qumran came from different individual animals, meaning they were separate copies, not pieces of one scroll that had broken apart. This diversity suggested the text was popular and widely copied by different communities, not the exclusive property of one sect. Even more revealing was the kinship analysis. By examining nuclear DNA markers, the team could sometimes determine that parchments came from animals that were related to each other. Siblings would share significant portions of their nuclear DNA. Parent and offspring would show characteristic inheritance patterns. Even cousins could sometimes be identified through genetic similarity. When the researchers found clusters of parchments made from closely related animals, it suggested those parchments were produced in the same location at roughly the same time. After all, closely related animals would have been raised together in the same herd or flock. Finding their skins used for parchment indicated a production center where multiple scrolls were being made from locally sourced materials. What the team discovered was exactly what you would expect if the scrolls came from multiple production centers rather than one isolated community. They found some clusters of related parchments, representing batches made in one location, mixed with completely unrelated parchments from different sources. The genetic diversity proved that the collection was assembled from multiple origins, not produced by a single community, copying texts for its own use. The implications extended even further when the team looked at specific biblical books. For the book of Isaiah, they found that different copies were written on parchments from different animals with no genetic relationship. This meant multiple communities were copying Isaiah independently, but the textual variance between these copies suggested different scribal traditions, different approaches to copying, possibly different theological emphasis. The DNA evidence supported what some scholars had suspected, but could not prove. The Dead Sea Scrolls collection represents multiple voices, multiple communities, multiple textual traditions, all preserved together in the caves near Qumran. The findings from the DNA analysis fundamentally changed how scholars understand the nature of the Dead Sea Scrolls collection and what it represents about ancient Judaism. If the scrolls were the library of a single isolated sect at Qumran, as the traditional theory claimed, then the textual variations would be difficult to explain. Why would one community preserve multiple conflicting versions of sacred texts? Why would they keep copies representing theological positions they disagreed with? It would be like a modern religious community maintaining multiple contradictory versions of their sacred texts in their library. Possible, but puzzling. But if the scrolls came from multiple communities across ancient Judea, then the diversity makes perfect sense. The collection is not a window into one group's theology. It is a snapshot of religious debate and diversity across Second Temple Judaism. Different communities had different versions of biblical texts. Different groups emphasized different aspects of religious law. Different scribal traditions approached the task of copying sacred texts with different priorities and techniques. The cow parchment fragments prove that texts were being physically moved between different regions. Someone brought scrolls written on cow parchment from wetter, more fertile areas into the Judean desert where they were eventually deposited in the caves. This demonstrates networks of communication, exchange, and possibly pilgrimage between different Jewish communities in the period before the Roman destruction of Jerusalem. The DNA evidence supported a theory that had been proposed by some scholars but lacked definitive proof, that the caves near Qumran were not the hiding place of one sect's library, but rather served as a geniza, a sacred storage place where Jews from various communities deposited old, worn-out or theologically problematic texts that contain the divine name and therefore could not be destroyed according to Jewish law. Jerusalem is only about 20 miles from Qumran. When the Roman army besieged Jerusalem between 68 and 70 CE, refugees fleeing the city might have brought sacred texts to the caves for safekeeping, adding them to materials that had already been stored there over previous decades or centuries. This would explain the enormous diversity of the collection. It represented the accumulated religious literature of multiple communities preserved together when the final crisis came. This interpretation explains features of the scrolls that the single sect theory struggled with. It shows why some texts are demonstrably older than the Qumran settlement. It reveals multiple competing textual traditions and a mixture of sectarian texts tied to one group's theology, alongside mainstream Jewish writings that any community might use. Most importantly, the Dead Sea Scrolls give us something even more valuable than insight into one radical sect. They give us a window into the diversity and richness of Jewish religious life in the Second Temple period. The crucial centuries when both Rabbinic Judaism and Christianity were emerging from earlier Jewish traditions. Genetic evidence illuminated the process of textual transmission in ways that purely textual analysis never could. Scholars could now track how different versions of biblical books were being copied, circulated, and preserved by different communities. They could see which texts were popular enough to warrant multiple independent copies and which were rare. They could identify which textual traditions had wide geographic distribution and which were localized to specific areas. For biblical scholars, this was revolutionary. It demonstrated that the biblical text was not monolithic or uniform in the Second Temple period. Different communities used different versions. There was not one universally acknowledged correct text that everyone followed. The textual tradition was fluid, diverse, and still evolving. The process of canonization, deciding which version of a text was authoritative, was still ongoing, contested and unresolved when these scrolls were copied. This has profound implications for understanding the development of both Judaism and Christianity. The diversity preserved in the Dead Sea Scrolls shows the rich theological ferment of the period when Christianity emerged from Judaism. The debates over scripture, law, messianic expectation, purity regulations, and community organization reflected in the scrolls provide crucial context for understanding the environment in which Jesus lived and in which the early Christian movement developed. The DNA findings raise new questions that could not even be formulated before. Now that scholars knew with certainty that scrolls came from different places, they could ask, can we identify specific production centers? Can we link certain textual traditions to specific geographic regions? Can we track how texts moved through ancient Judea and trace networks of religious communication between communities? Researchers are already pursuing these questions with expanded studies. They are analyzing DNA from more fragments. They are combining genetic data with other scientific techniques like multispectral imaging that can read faded or damaged text and materials analysis that can identify pigments and inks. They are building comprehensive databases of ancient animal DNA from different regions, hoping to create geographic reference maps that might allow them to pinpoint where specific parchments originated. The work continues, but the fundamental paradigm shift has already occurred. The Dead Sea Scrolls are no longer understood as the isolated product of one radical sect. They are recognized as a diverse collection representing the complexity and vitality of Jewish religious life 2,000 years ago. The DNA study of the Dead Sea Scrolls represents something larger than solving one archaeological mystery. It demonstrates how new scientific techniques can revolutionize our understanding of ancient texts and transform entire fields of study. For 70 years, brilliant scholars studied the Dead Sea Scrolls, using every traditional tool available. They made enormous contributions to our understanding. But there were limits to what handwriting analysis, textual criticism, and archaeological context could reveal. Certain questions simply could not be answered with those methods alone. Then molecular biology provided a completely different lens. By reading genetic information preserved in the parchment itself, scientists answered questions that had seemed unanswerable. They proved things that textual scholars had suspected but could not demonstrate. They provided objective evidence for theories that had been based on scholarly intuition. This interdisciplinary approach, combining traditional humanities scholarship with cutting-edge biological science, represents the future of archaeological and historical research. When experts from different fields collaborate, bringing different methods and perspectives to the same questions, breakthrough discoveries become possible. The scientists who sequenced DNA from the Dead Sea Scrolls did not set out to revolutionize biblical studies. They started with a simple conversation over lunch about whether genetics might help solve textual puzzles. By applying molecular biology to archaeological questions, they revealed truths that had been hidden for 2,000 years in the very material the sacred texts were written on. The Dead Sea Scrolls are even more remarkable than scholars realized. They are not one community's theological statement. They are a collection representing the diversity of Jewish thought during one of the most crucial periods in religious history. They preserve multiple traditions, multiple perspectives, multiple voices from different communities across ancient Judea. We only discovered this because scientists learned to read the story, written not in the text itself, but in the DNA of the animals whose skins preserved that text for 2,000 years. Scientists sequenced DNA from the Dead Sea Scrolls and were stunned, not because what they found was inexplicable, but because it revealed a story richer and more complex than anyone had imagined. A story written in genetic code that waited 2,000 years to be read.